Well, good morning, good afternoon, depending on where you are. Thank you so much for taking time to join us at Global Genes Live, a rare patient advocacy unsummit. I'm happy to welcome you to this session, Gene to Therapy, How Individualized Therapies Are Becoming Reality. My name is Alan Beggs. I'm the director of the Manton Center for Orphan Disease Research at Boston Children's Hospital, and I'll be your moderator for today's session. Before we get everything kicked off, I want to review a few brief housekeeping notes. First, all attendees are currently muted by the moderator to reduce any interruptions. We encourage you to use the chat function in the On24 window for any questions you may have throughout the session, and the moderator team will present them to me and our speaker. To open the chat, please click on the blue chat button located on the meeting controls at the bottom of your screen. If there are questions that we're unable to address or we run short on time, we'll do our best to follow up after the program. If you'd like to watch the session with closed captioning, please click on the white CC box to start the live captioning. Also, make sure to post your top takeaways and fun moments using Global Genes Live on social media and tag @GlobalGenes for a chance to be featured on our social media channels. The official event hashtag is hashtag 2020GG Summit. Finally, we encourage you all to participate as active listeners and take advantage of this opportunity to hear from our leaders and experts and ask questions of them. This program is your time to learn and find answers about critical challenges in rare disease research. So our expert panelist today is Dr. Timothy Yu. Besides being a close friend and colleague of mine, Tim is a neurologist and researcher in the Division of Genetics and Genomics here at Boston Children's Hospital. He completed his MD and PhD at UC San Francisco, followed by neurology residency at Massachusetts General Hospital and Brigham and Women's Hospital here in Boston. He's currently an assistant professor at Harvard Medical School, an associate member of the Broad Institute of MIT and Harvard. Tim works at the intersection of genetics, neurobiology, and bioinformatics, and is here to tell us today about some of his cutting-edge research on development of gene-specific therapies and how this fits into broader developments in the rare disease field. I'm now pleased to welcome our expert panelist, Dr. Timothy Yu. To kick things off, Tim, would you like to introduce yourself?
Hi, thanks so much, Alan. It's a real pleasure to be here. Thanks for hosting this chat. And a big thank you to the organizers for this chance to interact with all of you, if not in person, at least virtually. And I hope that you all learn or come away with some questions answered or maybe some ideas spun off. As Alan mentioned, I'm a neurologist and a geneticist at Boston Children's. I've been there 13 years now, and our lab has grown to go from thinking about rare diseases as trying to uncover the root causes, trying to identify the genes that are responsible for them. It's grown from that initial goal to now thinking about therapies, following actually along lines that Alan, you have developed for your rare disease interests in your group. But it's been an really interesting time to be in this space, and I hope to share some of that interest and excitement and challenge with you all today.
Great. Thank you, Tim. So just to give you the audience a quick overview, we'll have a little presentation. Tim will talk about his research and answer some questions. And then please do save up your questions and post them on the Q&A. And towards the end of the hour, we'll try to address everybody's questions. So we'll talk a little bit about the development of so-called N-of-1 treatments or individualized therapy. And Tim is going to tell us a little bit about some experience he has had, some really groundbreaking experience in this area. We'll talk about what it takes to kind of make a new drug from scratch, who you need involved in order to do this, and importantly, what are some of the safety considerations. But to kind of kick us off, Tim, I think you brought a video that kind of gives a really nice overview of what you've accomplished so far, and will set the stage for the rest of our discussion. So Maybe if we could run that video now, and then we'll get into the discussion.
There we go.
Right.
I'd like to start by thanking all of you for coming to help celebrate 2019's Rare Disease Day with us. I'm going to be telling you a little bit about a young girl named Mila. Mila was a typically developing child, precocious by many accounts, until around age 4, when she began suffering a mysterious decline.
She could have really been anyone's child. I think it's one of the reasons why Mila's story has gotten quite a lot of attention around the world. She went on hikes for 2-3 hours at a time. A group of people came up to her and they said, "Hey, little guy, aren't you cute?" And she said, "I'm not a guy and I'm not cute, I'm dangerous." You know, she was...
At that time, she was beginning to show some unusual symptoms, including pulling books up close to her face, having trouble discerning the letters in low light. At age 5, she began stumbling and unusual motor movements of her feet. Things became much more serious in a very short period of time.
The ophthalmologist, his advice to me was to chill out and just relax. That was 6 months before she lost her vision completely. And they did an incredible amount of tests that ended up in a diagnosis of Batten disease. No cure, no treatment, and it was 100% fatal.
We said, this is a family that's in need. We called it the family, and I introduced myself and I said we'd like to help. We had been thinking about how to roll out whole genome sequencing for patients for quite some time.
I will never forget the phone call I got. Our geneticist said, "Tim has something he wants to tell you." He actually thought outside the box.
The day that she received her first dose, I walked through the hallways of Boston Children's Hospital for a few hours. And I thought, oh my gosh.
Hi, Mila. Am I the big leader?
People told me that it was impossible. They told me that it would take 10 years and that it would never be in time for Mila.
This is sort of the best confluence of interests where, a family's desire to help their child also helps many other people in this very small rare disease community.
This was an entirely new drug targeted to one child. Mila's drug was called Milacin. It's named after her. This was an opportunity for me to tell Mila's story, to show the world that together we can race to create a new treatment path that could save the lives of children across hundreds or thousands of diseases. My mission, together with Dr. Yu, was to apply what we did for Mila to many other children.
We've seen a revolution in the way that we can make diagnoses. We can sequence genomes almost at the drop of a hat now. But to actually develop a therapy in this short period of time, that's a first. The question is, how do we scale this out to as many patients as possible in as safe a way as possible?
Why can't we have 1,000 drugs for 1,000 different kids? Mila is the first. She had to break that ice to say this is possible. Now we are entering another unknown territory, which is how do we rehabilitate a child like Mila that's lost so much?
It doesn't mean that everything is magically solved. It doesn't mean that every patient like Mila will have a customized drug for her tomorrow or the next day or next year. But it shows that when we put everyone together, it's possible.
I don't know if it's in time for Mila. It is too early to know that, but I'm really excited that we're in a day where it looks like there's hope.
Every time I see that video, it just tugs at my heartstrings again. So this is truly a remarkable story, Tim. I know that it was built on the work of a lot of people and many years of development and so on. But it's something that you accomplished in an incredibly and remarkably short time. So can you start to tell us a little bit, what does it take to actually make a new drug from scratch?
Thank you, Alan. I think I might preface my comments by saying that this was a really incredible and unusual experience for all of us. None of us reaching out to the family at the outset of this journey expected that we would be making a drug for her. That video shares a story which many of you may have heard before. I know it's been covered pretty broadly in the media, more so than many of us are used to. And it really began with a family reaching out for a diagnosis, looking for help, trying to figure out what was going on with their daughter. And it took us in many unexpected directions. So as far as, what does it take to develop a drug? I think the first thing I'd say is that it takes really good fortune. I think that I don't want to underemphasize that there are many things that lined up just so to make this story possible. And as I think I mentioned in the video, does this mean that this type of effort is going to be available for every child diagnosed with a rare disease? Not immediately, unfortunately. But it took a large number of things to make this new drug from scratch. And I'll just start by saying that one of the things that it took was a lot of basic research that had gone into understanding how genes are processed, how the instructions for making a gene's instructions turned into a protein, working out in exquisite detail how that happens. That was essential to developing a drug for this patient. And then the development of tools which allow us to change how that translation event occurs.
So Tim, are all mutations equally amenable to this type of an approach, or only some of them?
Right.
How do you decide if a particular patient has a mutation that could work or not?
Yeah, so we took advantage of a relatively unusual type of mutation in our patient, in Mila. Mila had a type of mutation that we estimate only about 1 in 10 patients with recessive genetic disorders have. And it was a particular type of mutation that changed the way that the genetic instruction set was assembled. It's a specialized type of splicing mutation that led to the introduction of a new splice site that joined one set of instructions to the wrong follow-up set of instructions. That genetic mistake caused by her mutation was something that we could fix. And it occurs in probably about 5 to 10% of individuals. But that's what we exploited in this particular case.
Mm-hmm. What about, for example, where the disease is present? So she had a disease that was affecting her brain. But what about liver diseases, muscle diseases, and so on? How does that impact this ability?
Right. I think I mentioned that many things lined up to allow us to make this therapeutic attempt. And one of the things that lined up was that our patient Mila had a really terrible disease, Batten disease. It primarily expresses itself. The symptoms are in the brain and in the spinal cord and in the eye. And we have at our disposal tools that work especially well in those places, that work in the brain, in the spinal cord, and in the eye. And so it was a good match. Her symptoms matched up with the type of drug technology we were using, something called antisense oligonucleotides, which are particularly well distributed. When you can inject it with the one needle injection, you can get an antisense oligonucleotide into those tissues efficiently, and they can work well. Other tissues are a challenge. Muscle, for instance, antisense oligonucleotides can work for muscle, but it's much more difficult. You have to give much higher doses because it's not as efficiently taken up. Other tissues like liver share some of the good properties like the brain and the eye. So liver, it turns out, is one of the places where you can give an antisense oligonucleotide and it's taken up efficiently. But then other places, other tissues, less so. So right now, the brain, spinal cord, eye, and liver are probably those that we have the best shot at using this type of technology. But other diseases that affect other areas, bones, kidneys, other organs, more work will be required.
How does this relate to gene therapy? Gene therapy is something that I'm sure a lot of people have heard about. And so we have oligonucleotides you mentioned and gene therapy. And what else is there?
Yeah, so that's an important piece to understand. So in general, therapies for rare disorders, that they can target different parts of biology. In traditional gene therapy, they target the actual gene itself, the gene that's mutated. And a traditional gene therapy is most typically replacing a gene that's not working particularly well with a new copy of the gene that you've provided, right? So you are swapping out like a spare tire, something new for something that wasn't working well before. In our patient's case, we were working with a therapy that didn't treat the gene so much. It actually treated the mutation. It was specific to our patient's mutation. It only works for patients who have that exact same mutation. Our patient had Batten disease, and there are many patients with Batten disease, but our drug only worked for her because she was the only one that we found so far who has her particular mutation. So that puts it in that special category of mutation-specific therapies, mutation-specific treatments. Finally, there are other treatments that can be effective for rare diseases that don't target the gene, they don't target the mutation, but they might target some other basic biology, like anti-seizure medications for genetic epilepsies. These are often small molecules that they don't go right after the root cause. They don't go right after the mutation or the gene, but they work to improve the way that neurons fire because they change the electrophysiology of the gene, of the brain. They affect channels, electrical channels. So it's important to sort of get those categories generally straight as you're leading to.
So those would be then considered personalized therapy, right? We've heard a lot about personalized medicine. If you, for example, know that you have a particular type of sodium channel mutation in the brain that causes epilepsy, then your particular condition would be amenable to a particular drug. But those drugs then work for more than one patient, presumably, right?
That's right.
So
yeah. So what we're seeing,
personalized medicine doesn't have to be N of one medicine, in other words.
That's right. Yeah. Personalized medicine is a pretty broad term, a pretty broad umbrella. And there are many different ways that a medicine can be brought to bear based specifically on how you are expressing the disease. But yeah, and so there are many types of therapies. Everything really, in a way, is personalized. A physician, a clinician working with the family is always going to choose the drug that he or she thinks is best for you as an individual based on all sorts of things. But this particular category, when you get down to the specifics of a drug that's made only for patients with one particular, with a particular gene mutated or a particular mutation in a gene, then those are much more exquisitely personalized or individualized than previous drugs.
Right. Okay. So in other words, what you're saying is an accurate genetic diagnosis is the basis for this type of an approach, that you need to get that gene test completed and successful, right? And
that's right. Yeah.
How often is that successful in finding a mutation?
Well, I think it's increasingly successful. And I think as you and many of us know who've been working in the space, in the gene discovery space, it used to be incredibly difficult. You have to really walk the planet to find special families that would allow you to map where a genetic disorder came from, and then to find the particular part in the genome where that was responsible for that disease. We live in a pretty remarkable time when we can sequence genomes at the drop of a hat. It's still not perfect. we have the technology now to be able to sequence genomes, and we're able to find diagnoses as a rule of thumb, roughly in about a third of cases that we try to go after, we're able to find a genetic cause for. But that number is steadily creeping up because we're learning more and more. But that's where it all starts. I mean, I think that in the old days, it used to matter the most to know, for instance, that you had epilepsy. And then after that, as our knowledge and our tools got better, it became important not to just know that you have epilepsy and you need anti-epileptic drugs, but it became important to know which specific gene was mutated. Was it a sodium channel? Was it a potassium channel? Was it something else? And now what we're seeing increasingly is that it's important to know what specific mutation you have, either what type of mutation you have, or in fact, what is the actual letter code of that mutation? Because the tools are getting good enough that you can take that information and match it with a therapy.
Right. Okay. So, and we've been talking about the underlying genetics and how that's important to know, and what the implications of that are for the type of therapy that gets developed. What about the medical condition itself and the stage at which somebody might get treated?
This is a really important point, and I think that it's important to have not just an understanding of the genetics, but where you are in the course of disease. And some genetic conditions, symptoms that as a family member or as an individual with that disease, symptoms that you care about might unfortunately represent water under the bridge. It might represent some problem that has to do with the way that, for instance, your brain was built in utero. Right? A particular genetic mutation that removes a critical circuit in your brain because it just doesn't form in the first place. Taking an individual with that type of condition and treating them after birth, it will never allow you to go back in time and fix that problem. However, there are, as you know, genetic conditions that don't affect the body's structure, but they cause a malfunction in the way that the body's working. And those are situations, those are types of diseases that a therapy applied even after birth can help. There are caveats to that, though, too. If there are genetic conditions where the malfunctioning is so severe that certain parts of the body have actually degenerated, the cells have died, then it depends whether you'll be able to bring that back. If you're talking about muscles, some muscles can regenerate, and you can develop a therapy that can allow muscles to properly regenerate. But if you're talking about the brain unfortunately doesn't have a lot of that capacity in it. The brain can rewire to a certain extent, but it can't regrow to a large extent. So, and unfortunately, we don't have ways to make that come back. So, picking the right disease, picking the right time in the course of the disease, and then picking your right goals, having a realistic sense of what's possible. That's also something that we all, as you know, think about.
Yeah, I mean, in the video, I guess it talked about how Mila had started to show symptoms, and then, tell us what happened with her over the course of time during which you were actually doing the laboratory work and developing the drug.
So this was a really key point that motivated us and our really big team to work very, very fast. We had met Mila at a time when she was losing the ability to walk, and she had almost completely lost her sight, but there was still a little bit, she still had some vision remaining. And she had lost many of her words, but she was still trying to speak. And Batten disease, her particular condition, is a really unfortunately aggressive condition. In the 10 months that it took us to meet her, diagnose her, and then come up with a therapeutic strategy, she suffered declines in all of those areas. Her walking went to requiring a walker. Previously, she was uncoordinated, but then she required a walker to be able to ambulate. her vision went from minimal to completely gone, and her speech continued to deteriorate. And so conditions like that really require the utmost in speed. And they're really tough, because it's hard to know when you have conditions like that. It's hard to know if any of that can come back. In fact, most of the time in brain degenerative conditions, when you lose some capabilities, that you generally don't expect them to recover. And so the goal for treatments is typically to slow the progression, ideally to arrest the progression, and improve quality of life to buy time. I think that that was a particularly tough one, but the fact that she was progressing during that time was also part of a huge motivation for us to move as fast as we did, and it actually allowed us to get concessions to the typical drug development program that otherwise wouldn't have been available to us.
Yeah. So actually, we'll get into some of the regulatory and safety questions in a few minutes. But clearly, I understand that could have a major impact. So with Mila, for example, then what was the outcome? What were the results that you were hoping to see with the treatment?
Well, when we started, it was such unknown territory. It was very, very difficult to know what to expect. In fact, I remembered that when we met with Mila's parents and sat down to discuss the possibilities, we had to be very, very open about the fact that this was so highly investigational that it was very difficult to know what to expect. We explained that some parts of Mila were probably irrevocably gone, that there were some parts of her brain that had already suffered injuries that were not recoverable, and that those wouldn't be coming back. What was unclear was that what parts of her brain had suffered injuries but were still recoverable, that they could actually recover from. And that was a big unknown. And then there were also, of course, healthy parts of her brain, too, that we were attempting to rescue or to save from any further neurodegeneration with our treatment. So from the family standpoint, they, of course, hoped for the best. They hoped for a cure to recover as much as possible. And we certainly had a long discussion about that, and they understood that a full cure was not realistic, but that stabilization was something that we could hope for. In particular, she was having a lot of seizures at the time that we started the treatment. And we were hoping that we could either delay the further progression of seizures or potentially improve the control of seizures that she was having trouble with at that time. And that part actually seemed to work. The most promising results of our treatment over the first years and then continuing now, it's been about 2.5 years, have been in the seizure response, that this medication that we had made for her under extreme duress really hugely diminished the frequency and severity of the seizures that she experienced in that first year. Right. It wasn't perfect. And some new seizures have cropped up recently that tell us that it's not a complete fix, but it did seem to help and continues to seem to help improve her quality of life from that standpoint.
Right. Well, so that's encouraging. So let me change tack a little bit here, and let's discuss what it takes to actually make this happen. So, as a physician, you have a clinical team, you see patients in the clinic, and it's pretty much you and some assistants and so on. And then in academic research labs, like both yours and mine, we have a staff of scientists, people with bachelor's degree, with undergraduate degrees in science, as well as PhDs or MDs. And we kind of work in this enclosed ecosystem in a hospital or a university laboratory. And we do basic science. So we work with cells and dishes, mice, and so on. But then when it comes to creating a drug, it goes way beyond that. So what does it take? Who does it take, and what kinds of people to actually make that happen? And how did you manage to assemble such a team?
Yeah, that's a great question. You're exactly right. Obviously, in an academic setting, there are certain things that we do very, very well. Academic medical centers were built to take care of patients and to do basic science. And the way that our system is built up, the machine that we have developed to develop drugs involves many moving parts that go way beyond the academic medical center. There are manufacturing teams whose only job it is to ensure a precise and safe drug supply. There's regulatory bodies, obviously like the FDA, whose job is to oversee how drugs are produced and made. There's huge teams devoted to ensuring the safety of the drug supply from a toxicology standpoint. So teams that are built to do nothing but test drugs, often in animal models, to assure that something may be safe for human use. And then, of course, there's the clinical trials team, which does live back at the hospital. But it's a huge industry and a huge effort. And much of that, a good chunk of that, lives outside of academic medical centers in the system. And in the case of Mielison, we had the chance to work with some amazing folks who stepped up from each of those domains. On the manufacturing side, we worked with a company, TriLink, which is in San Diego, that stepped up and said, we'd really like to help you and like to help your patient. We benefited from the volunteer advice of consultants, chemists, both independent consultants and some associated with companies like Ionis, who were in the field of making oligonucleotides, who offered us amazing advice as to how to make this type of work happen. We were able to find folks with expertise in regulatory affairs, beginning with folks at our own hospital who interact with the FDA on academic trials, but really rapidly extending to folks at the FDA, the Division of Gastroenterology and Inborn Errors, and as well as outside consultants like Lauren Black and Mary Pendergast and Sarah Goldkin, some folks who had experience at the FDA who are now offering their help to folks like us. And finally, we had the help of folks who had a lot of deep experience in how to run animal toxicology. These are physicians for small animals that really take exquisite care of their charges. They take care of mice and rats and monkeys and so forth, but their job is to make sure that that appropriate testing is done to the highest of standards. We worked with Julie Duville from Charles River Laboratory and a whole series of folks associated with her team to do animal testing in our case. It takes a huge, the cliche is it takes a village. It takes more like a small city, I think, to make these things happen.
Yeah, so I mean, this is what pharmaceutical companies do, and there's the old axiom: it takes 10 years and $1 billion to make a new drug, and it's because pharmaceutical companies kind of bring all this expertise together. And I'll just say, for the audience's sake, that that is still the primary path for development, and is extremely important. From myself and my research on myotubular myopathy, for example, I've partnered with a number of companies, both startup and larger pharmaceutical companies. And so I think one of the things we kind of want to bring out here is that, Tim, your experience was a real, very close, true partnership with one family and your research lab. And through the cooperation and assistance of a whole number of companies and people with regulatory experience, you were kind of able to pull this together. Another path, and probably a more common one, would be for academic scientists like us to develop something and then partner with pharma and biotechnology to kind of help bridge that development gap. And Global Genes obviously is an organization that kind of helps bring this all together and brings really what I consider to be the third leg of the stool, or the tripod, if you will, and that's the patients and the families, the patient community, because honestly, they're the people who know what they need. They're the people who help guide us in what to do. So we have more that we had thought to talk about, but there's also been a tremendous amount of interest and questions, I think, from the audience. So let's start addressing some of those questions. And actually, I realize there's a question here. How can patients help to harness efforts like this? Many patients have had whole exome sequencing performed. And is there a way for them to help bring this together for scientists like us and others to help analyze and act on?
That's a great question. In the case of Mila, we had the really remarkable fortune to work with an amazing family. Mila, her parents, Julia Vitarello and Alex Makovich, they had started a foundation called Mila's Miracle Foundation, and they're absolutely critical in catalyzing all of the work that we did. I think that families and their stories are really what all of this is about. The whole ecosystem lives to try to help these families, and it's incredibly motivating to have brilliant and motivated and energetic families like that to work with. We've certainly there's no lack of motivation in the field, and there are many patient communities out there that have done an amazing job supporting this work, or really being the focal point for the work. And what are the concrete things that they can do? Families and foundations can organize to put together patient registries, and really essential to knowing whether or not a treatment is feasible is knowing what the natural history of a condition is. And oftentimes for really rare conditions, that type of work, unfortunately, our system lets too much of that work fall onto the shoulders of families and foundations, but they do a heck of a job at it. We've worked with foundations like the AT Children's Foundation, which built a registry of some 250 patients and collected all of their mutations and put them into a open source repository that they made available to the community. And working together in that instance with folks like Anthony Philippakis at the Broad, they put all of that really incredibly valuable phenotypic information and whole genome sequencing information from their patients, and listing the mutations that were found in their patient population into one place where folks like us can review it and use it to assess whether or not a particular treatment option is feasible. So I think that's a really important thing to do, that families and their foundations that may be supporting them can help organize. Right. Other things that I think they also their voices are incredibly important in just also spurring everybody to act in this space. And I think that in interactions with the FDA, really, the FDA has gone from listening just initially, arguably primarily to scientists and physicians, to having the patient voice as part of the development process, to I'd say even more so recently, making the patient voice not just part of the process but the centerpiece of the process. I think that they deserve a lot of kudos for integrating that a lot more so than perhaps people might give them credit for. The voices are really, really powerful.
Yeah, thank you. Yeah, thank you, Celeste, for asking that. And as a follow-up, how can patient groups actually get researchers like us interested in researching their disease? What can they do to attract our attention? What do we need to know in order to know that we can actually make a benefit, can help?
I think that's a really good question. And Alan, you could speak to this too. I think in your position, running a laboratory working with many rare neuromuscular diseases. But I'll say for me, actually, I'll say humbly that education is where it starts. So there are over 7,000 rare diseases out there. And as much as we'd like to know each of them, no one knows them as well as the families themselves. And so we get outreaches from all sorts of families on a daily, weekly basis. And the first thing that I ask them to do is tell me more about your disease. I can read about it in a textbook, but to understand exactly what that experience is like, what the impact is on your family and you or your child's daily activities, really helps us figure out what are the realistic goals that are actually relevant for this population. Biology limits you in many cases as to what you can do, but the value of an intervention doesn't come from the biology, it comes from the daily life of the patient. And so if a family says that really, a family with difficulty with vision, it explains that being able to read a book in a classroom without a computer assistance is a sufficient goal to really enhance someone's quality of life, then that really helps us set our sights and our goals appropriately. And even though, we may be thinking, oh, I can't fix that, but I can't cure the disease. But if the family says, oh, you don't have to cure the disease, you can help us immeasurably by just doing X, Y, or Z, then that's stuff that we can work with.
Right. Yeah. Great. Improve quality of life. And so, from my own experience, and I would say that I've had people contact me by they'll Google the name of their disease and find that my laboratory website has a listing of that. Also, as I mentioned at the beginning, both Tim and I are part of the Manton Center for Orphan Disease Research. So Sarah asked about, Boston Children's Hospital, is this the only place that this is going on? And no, definitely not. We don't want to take credit for that. But here at Boston Children's, for example, the Manton Center for Orphan Disease Research focuses on patients with rare or undiagnosed conditions. And so we're allowed to enroll somebody without really knowing what they have and without even knowing exactly all the questions that we're going to be asking, which was kind of a paradigm shift a number of years ago, because the Human Subjects Protection Boards were requiring very specific questions. So you needed to know ahead of time exactly what you wanted to do. That's fine for common diseases, but for rare diseases, we don't know ahead of time. And oftentimes I'm contacted by families where we don't know what they have and they don't know what they have. So this provides a lot of flexibility. Ways to try to access this. Somebody asked the question, how do we ensure the democratization of personalized R&D? And how can you be advocates to ensure accessibility? Well, I think both Tim and I have emphasized how important it is for people to reach out themselves. So Mila's mom, I think she'd originally posted something on Facebook that you responded to. And through word of mouth via your wife, who's also a physician, you heard about this and responded. And I've had a number of families over the years reach out directly to me, sending me an email out of the blue, explaining their questions. You can find investigators like that, as I mentioned, by Googling the name of your disease or condition. You can look in the medical literature to see who's been publishing in this area, or ask your physician for help doing that. Ask your caregivers to help you in your journey to find the investigators. And oftentimes there may be one or two people somewhere in the world who've got a strong interest. In a few cases, I've had patients reach out to me and say, I've got X. Are you doing anything with it? And I've said, well, I actually know about X. I'm interested in X, but I've never done anything because we've never had any patients with it. So just making yourselves available, communicating, as Tim mentioned earlier, contributing to registries. With rare disease, it's very difficult to draw conclusions from just one or two people. But if we can assemble groups of individuals with similar conditions, then we can try to draw better conclusions. Tim, what about some of the risks and safety concerns that exist here? I mean, this is obviously all being done new and for the first time. And so what do we have to think about? And, how does the FDA and other organizations how do they help us manage these and ensure that we can do things in as safe a manner as possible?
Yeah, I think this is a really important point to draw out. Of course, the folks listening, those of you in the audience who are listening and contributing questions, those of us in the rare disease community, we're not at all strangers to risk, right? We know that many of our conditions are so rare that efforts that are being made on them are by necessity the first ever efforts for our particular conditions. And there's a sort of inherent sense that we have to take on a lot of risk because there are just not very many of us. Efforts that are looking very promising, like gene therapies, have well-publicized risks. And efforts like ours with Mealison for personalized medicine, they have truly even more risks because we don't know enough about this. It's such early days in this research that we have to be really careful to point out that there aren't a lot of data points. The data points that we've got are from, there are a couple of drugs in this same category as Mealison. Mealison was built as an antisense oligonucleotide, and we know that antisense oligonucleotides have some risks of liver and kidney dysfunction. In the brain or in the spinal cord where they're injected for neurologic diseases, there are risks of nerve injury that we are beginning to learn about. We'll have to continue watching the space to see how it develops. I think that this speaks to a point about what we can do to help make sure that, even though we acknowledge that there are these risks, what can we actually do to manage them? And I think more so than perhaps in any other time in recent history in drug development, collaboration is so important. As the rare disease community knows, there's a risk of fragmentation if efforts go into too many different directions and if data is not shared. And that's increasingly true if you're working with 7,000 rare diseases. Now, if you imagine not only are the diseases rare, but the drugs that you might be applying to them might be individualized or even down to one or a few patients, it becomes even more important to share data about how those experiences are going. If there are safety signals that show up for one patient, that has to be made accessible to other patients who are considering participating in research along these lines. So I think that we're going to have to see the development of structures that will help facilitate that type of sharing. In fact, I happen to be working behind the scenes on one together with Julia Vitarello, Mila's mother, to start a foundation that might have as its ambition a major mission to help catalyze the sharing of information so that this field can be developed responsibly. But I think efforts like that will be really, really increasingly important. They're already important now. We all know how important it is to break down walls for collaboration, but especially when we're talking about trying to make individualized drugs scalable and accessible, I think it's even more so important there.
All right, great. What about other, so you're one lab, we're one center. What types of other initiatives are out there to try to spread this? And actually, as part of that same question, one of our viewers asked, does this only children or what about adults? And we're a children's hospital, and that's kind of what we've been talking about.
Right.
But we don't have to only talk about children. We certainly don't want to.
Yeah, I think that so one, there is no inherent reason why the type of technology that we've been working with, that we've been discussing, there's no inherent reason why it can't also apply to adults as well. Though we're focusing on children primarily for a couple of reasons. One is that it is worth pointing out that it's such early days for this technology that we're only considering its deployment or its testing in the most severe genetic conditions. And just the way that this system, the biology works, that many of the most severe conditions do manifest in children. But that's not to say that there aren't very, very serious conditions, adult onset conditions that might merit consideration here too. So that, yeah, there's absolutely no restriction on that. With respect to other efforts, I think one of the exciting and rewarding things about the Nielsen case has been seeing the response to it. And there have been many foundations, and we've certainly received outreaches from many families and foundations who are interested in doing this themselves or asking us to help them do this type of effort themselves. And there have been many projects that actually that I know of but can't share details of, which are significantly down the path of trying to wield these tools to find something that might be a therapeutic option for their family or their disease. And there have even been industry recognition and industry support for this mechanism too. You know, Alan, you and I have the fortune of being associated with the NLORM Foundation. NLORM is a foundation started by Ionis and Biogen and a few other major industry players who Ionis and Biogen contributed hugely in the space to the development of antisense oligonucleotides because they were the primary scientific and pharmaceutical backers of Spinraza, the very first intrathecal antisense oligonucleotide that has had wonderful benefits for patients with spinal muscular atrophy. And they're sponsoring a foundation to further offer this type of avenue for other patients is one of the examples of the really encouraging developments and validations of this approach that we're seeing. I think I would say,
if I could just interject for a second, I think Christina from Ionis will be speaking at 2:10 in the oligonucleotide session. So anybody with more interest in that, definitely don't miss that session. Sorry, Tim, to interrupt.
I think that'll be a great opportunity to ask her questions about NLORM. It's a really promising early effort, and I think that I was just going to say stay tuned here. I think that there's likely to be more to come, and I think that this is the beginning of a trend that we've been seeing happen for a long time. That as our tools are getting better, we're being able to offer treatments with more and more specificity and to rarer and rarer conditions.
Getting back to actually the beginning of our discussion and the nature of the mutation and the genetic basis for a condition, there are actually a couple questions about autosomal dominant versus autosomal recessive, and I'll quickly address this a little bit, and then Tim, you can add on. But essentially, so if you have an autosomal recessive condition, you've got mutations in both copies of a particular gene, the one from mom and the one from dad. And usually, not always, but the great majority of the time, that results in what we call loss of function. So essentially, you're missing something. And so gene replacement therapy, where you use a viral vector to bring in a good copy of that gene, is essentially the act of putting back what's not there. And so that can be quite effective. In the case of Mila, you actually achieved that in a slightly different way by correcting one of her two mutations that was preventing the gene from being made properly, the protein, and actually allowed that one copy to make a full-size functional protein. And that's enough to obviously help her. But then in the question of autosomal dominant, sometimes autosomal dominant conditions can be caused by not enough of something. That's what we call haploinsufficiency. So there's insufficient levels of that protein. And there again, a replacement approach could work. But more often than not, it's creating something toxic, a so-called dominant gain of function, where it's actually creating a protein that does damage. And so then the question is, how do you take away that one bad copy or that 50% of the protein that's being made that's defective? And so oligonucleotides, if you can target them specifically to a particular mutation, can be used for that. Another approach that we're exploring with some of our muscle diseases is to actually flood the cell with more of the good copies. So you're not actually removing the bad copies, but by changing the ratio. So instead of having 50-50, maybe you'll have 80% of the good copy and 20% of the bad copy. That might be enough to actually help treat the disease. So it all really pretty much depends. We're coming to the end here, though. We've, I think, 3.5 minutes or so left to go, Tim. Do you want to just summarize, leave our listeners with any takeaways, maybe touch on, is this for everybody or not? And what does the future hold in your view?
Yeah, thanks for that opportunity. I think that one of the questions that we get, the biggest question that we get all the time after the news of the Mielison story has broke, is that is this a treatment that can be offered for a child? And are you saying that all drugs from this point should be made on an individual basis? And I would say that as excited as we are and proud as we are that we were able to do this, it still remains true that the general goal of a scientist is to make medicines that work as effectively as possible for as many people as possible. And we're not advocating. It's not that all drug therapeutic efforts will now be individually targeted and that everyone will be fragmented and fighting for themselves. That's not the point here. The point here actually is that on a case-by-case basis, and it depends on the gene, it depends on the clinical course, it depends on where you are in that clinical course, this is a new option. And if we think about it right, it offers a different way to think about it, a different paradigm. And on one hand, the drugs that you can develop using the Mielison approach are individualized, but on the other hand, they can be individualized in a way that makes the process the same every single time. And there are some examples in drug development history where getting the process right can be incredibly powerful, because if you, for instance, develop a process for making a vaccine, you can use it over and over again. For instance, for the different strains of the flu that come out every year, we're able to make vaccines that offer some protection against the flu, and that process stays pretty much the same. Or maybe a more cutting-edge example is some evolving stuff for cancer. You can make a vaccine that is tailor-made to a particular tumor that you may have, and that process it may be reliable enough that you can offer it for many patients at scale. So I think it's super early days. It's too soon to say whether or not that's going to happen, when that's going to happen. But I think that that's what motivates us to consider this approach, that can we develop a process for making individualized medicines that can be robust enough to offer to more folks.
Thanks so much, Tim. Tim, I think it's lunchtime. I hear your kids. Thank you so much for leading a wonderful discussion. And thank you to our audience for joining us today. And the pandemic has obviously led to significant changes in the way we do things. It's actually really nice that I think we can all kind of come together from wherever we are in whatever form we are and have these wonderful interactions. So thank you to everybody for listening. Please continue joining. At 1:10 in 10 minutes, please join the session as Gene Therapy Right for Your Disease, or Mapping Patient Inclusion in Drug Development. And at 2:10, the oligonucleotide session. So thanks to everybody, and thanks to the organizers very much for giving Tim and myself this opportunity.
Thank you so much.